All right. So the last thing we got is the scientific method. You should be able to put the steps in the correct order. And you should be able to tell me what's going on in each of these steps. Excuse me. This is how we gain knowledge through research, okay? And the great thing about the scientific method and with science in general, with greater technology comes different ways to test and determine how things function, or what causes, this leads to this, which leads to this, and understanding that, throughout the decades, we get a little bit more insight. And sometimes with that insight creates some confusion about what we thought was the gold standard or how we really understood things at one point in time, and how we get to this thing where we're like, well, I guess we don't really know how the hell some of these things function. Or the way we thought is totally turned upside down because of new technology. And understanding processes, how we understand these processes to determine what's going on, is that scientific method where you make an observation, you make an educated guess, you make some predictions and do an experiment, you collect data, you do a statistical analysis on it, and you write and form a conclusion. All right. So let's look at these things. The very first thing you have when it comes to the scientific method is you have to observe stuff. You have to interact with your world, right? It's one of those 6 characteristics of life, okay? Being able to interact with the world, seeing something, looking at a phenomenon, okay? Looking at some kind of event, all right? Where we use our senses to gather that information. Usually it's us observing something with our eyes. So we're just using that one major sense. Maybe we heard something, okay? You heard a sound and you want to figure out what that sound was coming from, okay? But we use our senses to gather this information, where we make an observation and we're like, huh, I wonder what this is. I think this is blah, 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 blah. That's a hypothesis, okay? A tentative explanation for what was observed, okay? It has to be something that's testable. If it's not testable, I can't prove it one way or another, okay? So it's gotta be something that I'm observing and I'm forming a hypothesis on that observation, and it's something that I can test, okay? Like, think of something, I don't know, gravity, okay? Why do I fall? Why do things fall down? Why don't they fall up? Do all things react the same? Does a feather fall at the same rate that a bowling ball would? Okay, and that's something that I can test. Make an experiment, okay? Come up with a series of procedures to test this hypothesis. Okay. There are several things that we need, and one of them's on this slide, and there's 2 of them on the next slide, okay? When it comes to an experiment, we have to have a good design for an experiment. What does that mean? It means I need variables, okay? And I need to come up with that variable that I'm testing for. Okay. Is that thing that I'm testing, is it a direct cause to what I'm hypothesizing? Or are there a number of other variables that could have affected the outcome? Okay. Is it a dependent variable? Or is it something contributing to it? Is it an independent variable? All right. That's what we're going to try to figure out. In any good scientific study, in any good scientific study that you read in a scientific journal, is going to talk about what they're testing for, okay? What the dependent variable is, all right? But then they're going to talk about all of the other potential variables that could have affected the outcome of the study. And that's just being a good scientist, okay? My hypothesis was this, and my experiment proved my hypothesis. I am the smartest man in the room, okay? But being able to kind of look at your data, look at your experiment, and see the potential flaws that could have been there during it, okay? Or something that you thought of afterwards, that's what makes the scientific method even better, okay? Variables, one thing, okay? You need a test group and you need a control group, all right? Because you want to see if that thing you're testing for is specifically directly causing this outcome. So you get a test group, and the test group is exposed to the experimental variable, okay? Your dependent variable, all right? And in that Labster lab called experimental design, this is what you're working on, this scientific method. And you have to figure out how, well, you get to make a test group and you get to make a control group, okay? And you have to be able to understand what a test group is. And the test group is what you're exposing to the variable, okay? And in there's a medication that you're looking at, trying to figure out, okay? A control group goes through everything, A control group goes through everything, but is not exposed to that variable, okay? And this is just to make sure that, that your test is valid, okay? And that the results are actually a direct result from what that thing is you're testing for, okay? Is that medication killing, I think, kidney cells in the Labster experiment? After you do the experiment, collect the data, okay? You observe stuff. Numbers are numbers, so hopefully they are objective. Hopefully you're not manipulating the data to help support your outcome. Obviously that can happen. It happens quite a bit. You see that right now with all the nonsense going on. But hopefully it is objective, and data is just the results of the experiment, okay? And you'll come up with some data for that, excuse me, Labster experiment. You put it in some tables, you put it in graphs, all right? And they'll show you, you'll have to do that, okay? It's pretty easy, just click some buttons, okay? And you can use that information in those tables and graphs to do some statistics and to make sure that there is some significance to those numbers, okay? You want to have a probability of less than 5%, okay? The closer you get to zero, the closer that specific variable was a direct cause to the outcome, okay? And that's what they're looking at here. They're trying to give you some examples there. See? Standard error, data points, come up with a standard error or standard deviation, okay? And you can see how statistically significant all the information is. The last thing we got here is with the scientific method is the conclusion, where you have a typed, written up analysis, interpretation of your data. Did it actually support your hypothesis? Yes or no? If it did, fantastic, awesome. You put it in a journal, scientific journal, okay? Your peers come out and rip you to shreds, okay? Or they'd be like, okay, this is sound science. And then somebody else will do the same. Follow your procedures and see if they get the same outcome. Okay? If your hypothesis was rejected, you'll go back and you'll make another hypothesis, and you'll do it all over again. And this is just a flow chart that kind of, puts it all together for you, okay? Flow charts are a fantastic thing to look at when you're studying, okay? Especially after you've reread the notes a couple times and kind of got a general gist of it. Just looking at this here and just being able to talk to your friend or your mom or your dad, whoever lives with you, okay? Or talk to your own head, okay? Being able to talk about this out loud. These flow charts kind of help to put all that information down on one little slide. After you do a number of experiments here, and all these experiments are starting to kind of add up, support each other here with every new experiment. With every new experiment that comes up with a positive correlation there, you can create some scientific theories and some scientific laws. Okay? And this obviously takes many experiments to get to that level of acceptability. Okay? And even those get questioned from time to time. Okay? Certain concepts, that scientific concepts that we believe we know, again, you may make a discovery. Okay? And it's like that turns our head upside down about how new species manifested. And we see that quite a bit because there's a whole kind of gap that we have in our family tree, in man's family tree, where we don't know how, Homo sapien got from some of the other rudimentary Homo structures. And it's fascinating and interesting. And I may try to put up a link to a video there where you can kind of see some of that and see how they've discovered just a handful of years ago a whole cache of human-like skeletons and how they fit in the grand scheme of things of our evolutionary tree. Disturbing, fascinating. It's fascinating, but the process of watching these tiny little people go through these tiny little holes in this cave system. Ugh. You'll have anxiety watching it. We have three basic theories here in biology that we're going to be looking at. Cell theory, number one right there. All organisms are composed of cells. New cells come from pre-existing cells. So we're going to be looking at cells pretty significantly throughout the first, 3/4 of this class. Homeostasis is another basic theory here. And, this is how the human body functions, okay? This internal environment maintained within a specific little range, and we have a number of homeostatic mechanisms within the body. Come to 109, come to anatomy and phys, or come to physiology class, and we dive into all these, okay, in much more greater detail. And then obviously evolution, okay? Obviously evolution. There's another kind of one, they refer to it as the central dogma. And the central dogma is basically a theory to how we get from DNA to structure. Okay? And we'll talk about that when we talk about how mitosis and meiosis occurs and how we read the genetic information that's in our nuclei and how that whole thing happens. And that's what's called the central dogma of biology, which is basically this group of theories kind of clumped together here, almost like a law. The last couple things here we'll leave off. technology, man. Okay. There's a lot of challenges that face science. Again, science is just a systematic way of trying to look at something and have a better understanding or better knowledge base on how something functions within the natural world here. And technology has increased our ability to understand a lot of processes a lot more effectively. But in that process, we've also created a whole host of other new questions because of that technology. The book also goes into this concept of how does technology affect mankind? And we're not going to get into that as much, and we may hint at some studies later on when we talk about some concepts, especially in evolution, genetic engineering, stuff like that, when you get into mitosis, meiosis. So we'll maybe hint at some of that. But that's what they're trying to talk about here. And they kind of do an introduction of this word called biodiversity. And understanding what the word biodiversity is really all you really need to do at this point. We're going to get into it much more later on when we talk about ecosystems and we talk about evolution. Biodiversity is the reason why you want ecosystems to thrive, okay? You want them to have an abundance of various different creatures, species, okay? And all of that variability here, it's on the terms of genomes, of genes, okay? If we have a lot of species, we have a lot of genes. And if we have a lot of genes, we have a lot of potential adaptability that can happen within that ecosystem. The fewer and fewer and fewer species that you have within an ecosystem, that just means less genetic information. So that means that there's fewer genes for which to create adaptations, right? And it creates these things called bottlenecks because of that. And with species that are on, the endangered species list, we see that phenomena. We only have a handful of species here. Well, that creates this huge bottleneck because we only have a handful of genes to create more and more and more of that individual species. Eventually the genes, the gene pool is not diverse anymore and the offspring are not viable. And it all goes back to this concept of habitat loss and biodiversity. You want an environment to have a huge number of species so we can have a lot of adaptations. Right? If not, species go out. They die, they extinct, and you can see there's estimates that about 400 species per day are lost due to our activities with habitat loss. Okay, coral reefs, tropical rainforest, those are the two major ones there. Okay. I can't remember there's even a number there's even a crazy number associated out there with the number of species we just potentially don't even know about because of habitat loss or because habitats are so diverse and so massive and so huge. It's kind of hard to go through and truly get a sense of being able to catalog every single one of these species here. These habitats provide a whole heck of a lot of stuff. Food for the big one, okay? Food for us, but food for other animals to feed on and eat on and survive and advance. And if you just remove one of these species from within that kind of web of the ecosystem here, an ecosystem can start to falter and species start to die off. And that's what they look at here over these two slides. Okay? Food, medicines. There are a number of different medicines that we've derived from various different plants, animals, okay? Obviously plants, it's a huge one, okay? But, even blood from the horseshoe crab, they've utilize that for different ways to handle, medications for a patient during a surgery. Okay, just the raw material loss. Okay. Some other questions here associated with science, emerging stuff. Obviously, COVID would be on this slide now. Okay. Where do these emerging, to me, this is the most fascinating thing, right? Besides coming from a nefarious lab somewhere, where do these things come from? Are they just from being exposed to various different animals all herded together, or insects all kind of herded together in a spot? Do we speed up adaptations that way? Change in our behaviors? Look at how this thing started overseas in China. And just because of the way our economy works, the global economy works now with international travel, it gets to Europe really fast, to Australia pretty fast, to the United States really fast. That's a change in behavior. That wasn't necessarily the case 100 years ago or 200 years ago even. Use of technology. Legionnaire's disease is the example. Legionnaire's disease caused by a bacteria that resides within large air conditioning units. Okay, so obviously the use of technology, the creation of technology has created something in the process. The globalization. Okay, I talked about that with changes in behaviors. Okay. Mutating. The biggest problem with this, guys, and having these different types of bacteria that are immune to various things, not utilizing medications properly. Instead of taking all 17 pills, you may take 7. Start to feel good. Okay, I don't need to take them. Instead of taking all 15, kill off 90+% of them, 99% of them. Now we've left a bigger number around, and those were exposed, and now they can create a immunity to that medication. And that's what we've seen with a lot of antibacterial medications. That's why we have a number of bacteria that are superbugs. But this is a fascinating question, okay? Looking into some of the reasons why we have these emerging diseases, that made me a good kind of topic to kind of research and figure out. And then obviously, another thing that science is perplexed with is this concept of climate change. Okay? There are changes that happen within these various different cycles that we'll talk about at the end of the semester when we look at chemical cycles. Okay? And the extent to which man has contributed to these. Okay? Even though there's a natural cycle that has existed, and there's data to show that, man has definitely sped this up and increased a lot of these things. Okay? And that can't be denied. And so climate change is one of those things that dilemmas that science is trying to deal with to try to figure out exactly kind of what our role in this unfortunate inevitability has caused.